Crohn's disease is a chronic disease that primarily affects the digestive system. It is caused due to an abnormal immune response that causes the inflammation of the intestine walls. There are a wide range of different symptoms associated with this disease, from abdominal pain till diarrhea. However, the principle and the basis of this disease is not fully understood yet. It is thought to come from a combination of genetics, abnormal immune response that triggers cell signaling, and environmental factors. Let's see how it works.
The human body is colonized by trillions of microorganisms referred to as the human microbiota, which colonizes the skin and mucosal body surfaces of living organisms. These microbes inhabit competitive and complex environments and deploy different ecological strategies for survival. In the gut, more than 500 types of bacteria are present, which are usually categorized as either good or bad by receptors in the epithelium. But unfortunately, in people with Crohn's disease, these receptors don't work properly and lead to a weakened mucosal layer, which leads to the inability to clear bacteria from in the intestinal walls, which also results in a depletion of the healthy human microbiomes and an increase in potentially pathogenic ones. Interactions between the intestinal and cell pathogen recognition receptors and microbial ligands trigger signaling pathways associated with the immune system.
The problem with Crohn's disease is that this weakened mucosa located in this area can allow the proliferation of potentially harmful bacteria. So this is the case of the adherent invasive E. coli. This set of bacteria, when they are proliferated, they can be translocated, and then the macrophages will engulf them. Every time they engulf them, they're going to produce one tumor necrosis factor alpha signal that will trigger the inflammation. So what's the problem in Crohn's disease? The problem with Crohn's disease is that these bacteria can't be killed by the macrophage. So the moment they can't be killed, there's going to be proliferation inside of it. Consequently, we're going to have a permanent signal of the tumor necrosis factor alpha, and this is going to produce a constant inflammation. So, this is the problem. How are the pharmaceuticals nowadays trying to face this problem? They're producing new drugs, such as infliximab, and they're acting on this step of the pathway. If we can't deliver the signal, we're not going to get the inflammation. However, this is not efficient, and we can't solve a problem like this. What we suggest is solving the problem from the very beginning, and avoiding and preventing the bacteria from translocating through the epithelial cells. Our idea is based on immune stimulation, focusing on the prevention of bacterial translocation by enterocytes. So we want to reinforce the mucosal barrier using a self-modified probiotic, which will prevent translocation from happening. We had a problem,
And we found a solution.
and now we are ready to make a difference.